Cell structure and organization. In today's crash course, we'll be talking about prokaryotic versus eukaryotic cells, light and electron microscopes, cell organelles and compartmentalization, introduction to protein synthesis, adaptations of cell structures to functions, and lastly, organizations in a multicellular organism. Before we get into the differences between prokaryotic and eukaryotic cells, what are cells? Cells are the building blocks of life. They are the simplest unit that can live independently in unicellular organisms. In multicellular organisms, they are the basic unit of structure and function in an organism. Prokaryotic cells are also known as prokaryotes, and eukaryotic cells are also known as eukaryotes. Similarities between these two types of cells include the presence of cell membranes, cytoplasms, ribosomes, chromosomes, DNA, and mitochondria. However, differences between these two types of cells include that firstly, in prokaryotic cells, DNA is not enclosed within a membrane. This means that there is no nucleus. Membrane-bound cell structures, cell organelles, are absent in prokaryotic cells as well. This means that prokaryotic cells do not have an endomembrane system. Prokaryotic cells are generally smaller in size. In eukaryotic cells, the nucleus is bound by a membrane known as the nuclear membrane. This means that the DNA in the cell is stored in the nucleus. Eukaryotic cells also contain membranous cell organelles, such as the mitochondria and chloroplasts. In eukaryotic cells, there is also an internal membrane system known as the endomembrane system. The nucleus, endoplasmic reticulum, Golgi body, lysosomes, vesicles, and vacuoles are all enclosed within the internal membrane system. Lastly, eukaryotic cells are larger in size than prokaryotic cells, and they are more complex. So what exactly are prokaryotes and eukaryotes? Unicellular organisms such as bacterial cells and archaea are considered prokaryotic cells, while animal cells, fungi, protists, and plant cells are considered eukaryotic cells. Next, we'll be talking about microscopes. Cells are very small in size and therefore require the help of light or electron microscopes to visualize them. Cells are small because the small size allows the cell to have a large surface area to volume ratio, which allows diffusion of materials in and out of the cell quickly. Small cell sizes also allow molecules to be transported over short distances, which helps to speed up many cell activities. Microscopes differ in their magnification and resolutions. These refer to the minimum distances between 2 points at which they can be seen separately rather than a single blood point. It is also the ability to distinguish between two objects. It determines how clearly a small object can be viewed. The total magnification used is the product of the eyepiece magnification times the objective light microscopes versus electron microscopes. Light microscopes are less powerful than electron microscopes. Light micrographs can come out as colored images, whereas electron micrographs cannot. Cell structures that can be seen under a light microscope are the nucleus, cytoplasm, cell surface membrane, cellular cell wall, chloroplast, and large central vacuole in plant cells, or multiple small vacuoles in animal cells. For electron microscopes, there are two types, namely the scanning electron microscopes, SEM, or the transmitting electron microscopes, the TEM. Organelles that can be seen under electron microscopes are the rough and smooth endoplasmic reticulum, Golgi bodies, vesicles, mitochondria, and ribosomes. Viruses and individual atoms also can only be seen under the electron microscope. Animals versus plant cells. Cell structures found in both animal and plant cells are the nucleus, nucleolus, mitochondria, cytoplasm, cell membrane, rough and smooth endoplasmic reticulums, ribosomes, Golgi bodies, and lysosomes. However, animal and plant cells differ in that firstly, animal cells do not have a cellular cell wall, therefore they may burst easily. In contrast, plant cells have a cellular cell wall. The cell wall provides mechanical support, which reduces the chances of the plant cell bursting. Animal cells do not have chloroplasts, whereas plant cells do. This is as plant cells undergo photosynthesis and animal cells do not. Centrioles are present in animal cells, while they are absent in plant cells. In animal cells, the storage carbohydrate is glycogen granules, whereas in plant cells, it is starch granules. Cell organelles and compartmentalization. The cell organelles in a normal cell are the nucleus, cell surface membrane, cytoplasm, cell wall, chloroplast, vacuole, mitochondria, endoplasmic reticulum, Golgi body, and lysosomes. Before we look at these organelles in detail, we first need to understand what is compartmentalization. Compartmentalization refers to how the cell keeps structures and processes of different functions separate. Now onto the different cell organelles. Firstly, the nucleus. The nucleus has two membranes. Its functions include controlling cell activities such as cell growth and repair of worn-out parts. It is essential for cell division, and cells without a nucleus are unable to divide. This is as the nucleus contains genetic information in the form of DNA, which is deoxyribonucleic acid, together with histone proteins, forming chromatin and chromosomes, which is stored in the nucleus. DNA molecules consist of sequences of nucleotides, genes, which contain the chemically coded instructions for synthesizing proteins needed by the cell. Thus, the nucleus is able to detect what and how the cell will conduct processes. The nucleus is one of the organelles in the cell that is bound by a double membrane system, which is known as the nuclear envelope or nuclear membrane. Within the nuclear membrane, there are nuclear pores, which are proteins that form channels between the nucleoplasm and cytoplasm. The nuclear envelope is also continuous with the rough and smooth endoplasmic reticulum. Within the nucleus, there is a structure known as the nucleolus, the plural of which is nucleoli. The nucleolus is not enclosed by a membrane. It is where ribosomal subunits are synthesized. The nucleolus contains rRNA, and proteins from the cytoplasm enter the nucleolus and are assembled together with the rRNA to form ribosomal subunits, which will then form ribosomes crucial in protein synthesis. Next, the cell surface membrane. This is also known as the plasma membrane. It is a bilayer made up of modified lipids or phospholipids. The bilayer structure. The phospholipid bilayer is made up of many phospholipid molecules and is 2 phospholipid molecules thick. It separates the intracellular from the extracellular matrix. Within the bilayer, there are protein channels embedded, which aid in the transport of substances through the membrane. The phospholipid molecules are arranged in such a way that protects the hydrophobic core from the liquid in the extracellular or intracellular matrix. This way that the molecules are arranged can be represented by the fluid mosaic model. Fluid refers to the fact that the molecules can move about freely and can switch positions laterally. Mosaic refers to proteins embedded and scattered about randomly within the membrane. Each phospholipid molecule is made up of a phosphate group, a glycerol molecule, and 2 fatty acid molecules. In a non-modified lipid, 1 glycerol molecule reacts with 3 fatty acid molecules in a condensation reaction to form 1 single lipid molecule. However, in phospholipid molecules, the phosphate group replaces one of the fatty acid chains. The byproduct of this reaction is a water molecule. Next, the cytoplasm. The cytoplasm contains the cytoskeleton. Microfilaments, microtubules, and centriosomes are all proteins within the cytoskeleton that reinforce the cell. The cytoplasm contains 90% water and 10% dissolved solids. This allows many chemical reactions to occur because of the water being present. The cytoplasm also contains cell organelles such as the chloroplast, vacuoles, mitochondria, endoplasmic reticulum, Golgi body, ribosomes, and lysosomes. The cell wall. First of all, the cell wall is fully permeable. It is made up of multiple layers of cellulose and is only found in plant cells. The functions of the cell wall includes providing mechanical protection to prevent bursting of the plant cell. Essentially, the purpose of the cell wall is to shape, support, and protect. Now, the chloroplast. An important structure in the chloroplast are thylakoid discs. These discs form stacks known as granum or plural grana. They are membrane-bound structures that contain chlorophyll pigments. These are crucial as they capture light energy, which is converted into chemical energy, glucose, during photosynthesis. Chloroplasts may also contain other yellow and orange pigments known as carotenoids. Chloroplasts are found in plant cells only. Animal cells cannot conduct photosynthesis. Chloroplasts also contain small amounts of DNA. The fluid-filled space within the chloroplast is known as the stroma, which contains enzymes essential for photosynthesis. Now, vacuoles. Vacuoles in plant and animal cells are very different. Plant cells have a single large central vacuole containing plant cell sap. This is made up of approximately 90% water with dissolved substances like sugars, amino acids, mineral salts, and waste. The membrane of the vacuole in plant cells is termed tonoplast. Animal cells have multiple small vacuoles, which usually exist temporarily in the cell. These vacuoles are only formed when the cell needs them. Vacuoles usually have a variety of functions and are usually used for storage of substances within the cell. Now, let's talk about the mitochondria, the powerhouse of the cell. The mitochondria is the site of aerobic respiration, during which food substances are oxidized to release energy. The chemical energy in food is oxidized and converted into ATP molecules. ATP stands for adenosine triphosphate molecules, which provide energy for all cell activities. Essentially, they are like the currency that drives the cell. The mitochondria is a double membrane-bound organelle, and it has an outer membrane and an inner membrane. The outer membrane is smooth, while the inner membrane has numerous folds on it. These folds are called cristae, or crista in the singular form. This increases the surface area to volume ratio, which in turn increases the surface available for chemical reactions to occur. This allows the membrane to accommodate more electron transport chains, leading to faster aerobic respiration. The compartment between the inner membrane and outer membrane is also called the intermembrane space. And the compartment enclosed by the inner mitochondrial membrane contains many enzymes, together with very small amounts of DNA similar to chloroplasts. Note that cells with high energy requirements generally have larger numbers of mitochondria, as they have greater needs for aerobic respiration. For example, cells that require active transport have large numbers of mitochondria. The endoplasmic reticulum, or ER for short. The endoplasmic reticulum is a partially permeable membrane system. It is a membrane-enclosed passageway for transporting materials. It is continuous with the nuclear envelope, and the internal space of the ER is known as the ER lumen, which contains enzymes that catalyze many different types of reactions. There are two types of endoplasmic reticulum, the rough ER and the smooth ER. On the rough ER, the surface appears rough due to ribosomes attached to the outer surface. The smooth ER has a tubular appearance and looks smooth as ribosomes are absent. The rough and smooth ERs actually have very different functions. The rough ER contains ribosomes, which are the sites of protein synthesis. The ribosomes synthesize and package proteins. The membrane of the rough ER can thus transport proteins made by the ribosomes to the Golgi body by forming small transport vesicles. The function of the smooth ER is to synthesize substances such as fats and steroids. The smooth ER also converts harmful substances into harmless materials through detoxification. For example, there are large amounts of smooth ERs in liver cells which function to synthesize and process cholesterol and other lipids, and serves as a major site of detoxification. Note that ribosomes found on the surface of the rough ER produce extracellular proteins to be exported, while free ribosomes in the cell produce intracellular proteins. The extracellular proteins are exported as the protein's functions are needed outside of the cell. For example, hormones and digestive enzymes. The Golgi body. The Golgi body is also known as the Golgi apparatus or the Golgi complex. It is not physically connected, but connected through vesicles. The Golgi body consists of a stack of flattened membrane-bound bodies, also known as cisternae. The membrane system is not continuous with the nuclear envelope, unlike the endoplasmic reticulum. Vesicles can be seen fusing with one side of the Golgi body, the cis phase, and pinching off from the opposite side, the trans phase. Vesicles are small membrane-enclosed sacs formed by budding from the membrane of another organelle. They function to carry materials from one organelle to another. The function of the Golgi apparatus is to chemically modify and package proteins made by the rough ER into forms the cell can use. Packaged proteins then live in vesicles for secretion out of the cell. These vesicles are now called secretory vesicles. The Golgi body does so by folding the proteins or adding other materials such as lipids or carbohydrates. Now, lysosomes. Lysosomes are small sacs of digestive or hydrolytic enzymes that are dispersed in the cytoplasm. These enzymes usually function optimally under acidic conditions around pH 5. They take in damaged and worn-out cell parts. They are membrane-bound, and they turn cell waste into building material. Introduction to protein synthesis. Protein synthesis is quite complicated, so I'll be explaining more in depth in another video soon. Essentially, the process of protein synthesis starts with DNA, which is transcribed into mRNA, which is messenger RNA. The mRNA then leaves the nucleus and heads to the ribosome in vesicles. In the vesicles, translation occurs where ribosomal subunits attract tRNA molecules, which are transfer RNA molecules. A long chain of amino acids then emerges as the ribosome decodes the mRNA sequence into a polypeptide. The protein formed then lives in a vesicle, and a new protein is synthesized. Adaptations of cell structure to function. Today we'll be looking at 3 case studies. Firstly, root hair cells. The main function of root hair cells is to absorb water and mineral salts from the soil. The first adaptation is the elongated extension, which is the root hair. This increases the surface area to volume ratio of the cell, which allows more efficient absorption of water and mineral salts from the soil. The cell also has an extremely large vacuole, which allows more water and mineral salts to enter the cell. The cell has a very concentrated cell sap, which creates a greater water potential gradient between the cell sap and surrounding solution in the soil, allowing water molecules to be absorbed more quickly via osmosis. There are large numbers of mitochondria in the cell, as energy released during aerobic respiration is used for active transport of mineral salts into the cell. There is an absence of chloroplasts, as these are unnecessary organelles. Root hair cells are found underground where sunlight is absent, so photosynthesis cannot occur. Secondly, xylem vessels. The main functions of xylem vessels are to provide mechanical support to the plant and to transport water and mineral salts from the roots to the leaves for photosynthesis. The first adaptation is lignin, which is a rigid and waterproof substance deposited on the cell walls of the xylem vessels. Lignified walls of the xylem strengthens the vessels and prevent them from collapsing. When they are bundled together, they provide mechanical support to the plant. Xylem vessel cells are hollow, with no cross walls or protoplasm. This means that xylem vessels are dead. This enables water to move more easily through the central space or lumen of the xylem. Xylem vessel cells are also continuous, from the roots to the stems to the leaves. This allows water to be transported from the roots up to the stems and to the leaves to be used for photosynthesis. These are the 3 types of lignification patterns, with the youngest being annular, then helical, and pitted being the oldest. The pitted pattern is the strongest, as it has the most amount of lignin deposited on the cell walls. Lastly, red blood cells. The main function of red blood cells is to transport oxygen from the lungs to the organs of the body. The first adaptation is the presence of hemoglobin, which is a red pigment. Hemoglobin binds reversibly with oxygen to form oxyhemoglobin, which helps to transport oxygen in the red blood cell around the body. The second adaptation is the lack of a nucleus. The absence of the nucleus allows more hemoglobin to be packed inside the cell, which means more oxygen can be transported. The red blood cells are biconcave in shape, which increases the surface area to volume ratio of the cell, thus allowing more efficient diffusion of oxygen to the center of the cell. The cell membrane is also elastic and flexible, which allows the cell to change its shape into a bell shape while squeezing through capillaries. Lastly, we'll be looking at the organization of a multicellular organism. Firstly, tissues. A group of similar cells working together to perform a specific function is called a tissue. There are 4 types of tissues. Connective tissues function to join the different tissues of an organ together. Epithelium tissue consists of epithelial cells working together to protect the body and organs from injury. Muscle tissues consist of muscle cells working, contracting, and relaxing to bring about movement, and nervous tissues consist of nerve cells, neurons, working together to coordinate and respond to changes to stimuli. Simple tissues are tissues that contain cells of the same kind, for example, the epidermis or mesophyll tissues in leaves, whereas complex tissues have more than one type of cells, such as blood, xylem vessels, or phloem vessels. Organs. Organs are a group of different tissues working together to perform a specific function. For example, the heart works to pump blood around the body. The stomach churns food and digests protein. The liver functions to store glycogen and deaminate amino acids and conduct detoxification. The brain controls and coordinates the activities in the body, and the lungs conduct breathing and exchange of gases. In plants, the roots, stem, leaf, and flower are examples of organs. In the leaf is made up of the mesophyll tissue, xylem vessel tissues, and phloem vessel tissues. The mesophyll tissue is involved in photosynthesis, while the xylem and phloem vessels are involved in the transport of substances. Systems. Systems are defined as several organs working together for a common purpose. Various organ systems work together to make up the entire organism. For example, the digestive system functions to ingest, digest, absorb, and assimilate food. The circulatory system functions to transport substances around the body. The excretory system functions to remove metabolic waste from the body. The respiratory system functions to absorb and remove respiratory gases from the body. The nervous system functions to coordinate and respond to stimuli. The reproductive system functions to make gametes and produce offspring. In plants, the plant body consists of the shoot system and the root system. Functions of the shoot system are photosynthesis, reproduction, storage, transport, and hormones. Functions of the root system are anchorage, absorption, storage, transport, and hormones. That's all from me. Thank you for watching.